Thank you very much for that kind introduction, and I of course want to thank the organizers and the sponsors of this meeting. It's a very difficult task to put all this together and to keep everyone together, and I'm happy that so many people stayed all the way to the end. Hopefully I can produce a good kind of summary of what I've been doing and maybe some perspectives on what we all should be doing next. So I'm going to be talking today on the transport and retinal capture of the macular carotenoids, and this project has been a particular passion of mine for a long time, 10 or 15 years now. This is a long-term project. This is, as we'll see, is basic biochemistry. And it's very challenging. And in part, some of the challenges come from this is that we're dealing with, if you're trying to solve the biochemistry of how this works in the human eye, we're left with very few animal models. We know that carotenoids behave very differently in other animals, and in a lot of the things that we're looking at in the macular pigment and the macula lutea is that the macula lutea, the yellow spot of the retina, is unique to primates. So primates do things differently. So you can't do, if you're going to try to figure out the metabolism processes, mice are not so good, rabbits aren't so good. You're really either working with humans or monkeys, as we just heard this wonderful talk on monkeys. And the work that was done, that was sponsored by Roche and DSM, was a long-term project, presumably very, very expensive, very challenging to do. And the numbers that we just saw, I just want to caution, Toss, when he was looking in those numbers, you're looking at a couple monkeys there. Yeah, so so you can't do statistics, but the fact that they did it, that's how monkey experiments are done. I've got some researchers in my department who work on monkeys, and one or two monkeys keeps them busy for a year, and they're happy if they publish one paper a year on that monkey. It's very different than in other specialties. So anyway, just to address the problem that we're looking at is we all know here that lutein and zeaxanthin form the macular pigment. That's a given. That's been known now, and beautiful work that's been known for decades here. And we've also heard multiple lines of evidence that indicate that lutein and zeaxanthin are very likely to protect against age-related macular degeneration. They may also be important in cognition, as we've heard about. They may be important in the development of the macula itself. Which comes first, the macular pigment or the fovea? Or the fovea or the macular pigment. We need to try to understand, because they seem to go together. If you don't have a fovea, and some humans don't have foveas. For example, people who have albinism do not have foveas. They don't have macular pigment. So there are a number of things that need to be looked at. They tend to go together with this kind of primate fovea. And we know that the dietary sources of all of our lutein and zeaxanthin that are shown here in the classic picture from Max Snodderly all have to come from our diet, from orange yellow fruits and vegetables, green leafy vegetables. And so when I was working on this, started working on this project in the early 1990s, I saw pictures like this in the literature. I saw as a clinician, bringing in my clinician hat of this, I saw as a clinician fixing a retina detachment. There was that yellow spot on a detached retina telling me exactly where the fovea was. Clearly, the nature had gone out of its way to make the macula yellow and to really concentrate this. So I wanted to understand how this process could be happening. And you can see here in the classic picture here, it's right in the foveal pit here, right here. So this is, the vitreous is here, the top of the eye is up there. Just to show on the anatomy, this is the retinal pigment epithelium back here. And you can see there's the photoreceptor layer here, but the outer segments, which are not shown very well here because it's kind of smeared on this picture, are out here. They're relatively xanthophyll free. It's really the structures more of the inner retina, and these are the inner segments of the cones, and which is responsible mainly here, the inner segments going into the Henle fiber layer. These are the axons of the cones running out here. And you see it in the inner plexiform layer also. So this is a very specialized structure here, and it's very specialized concentration in one part of the eye. And the specificity, as we know, is extraordinary. we know that there are over 600 carotenoids in nature. For those of you going on to the Poland meeting, I think the count is actually probably more like 850. It's getting close to 1,000. So there's an enormous number of carotenoids found in nature. And we don't eat everything in nature, of course. And a typical human eats in the range of about 50 different carotenoids. So there is some degree of selectivity just by our selection of what we eat. But there is another level of selectivity is that not everything that we consume gets absorbed. Some of it may get damaged or metabolized in the digestive system, as we heard. Probably even the esters that we have are getting cleaved probably either in the gut or as they're being absorbed from the gut. Some of the various epoxides may be metabolized also, and some just simply are ignored. There is a level of selectivity, probably through scavenger receptors is what it's thought. I'm not an expert on this, but at the intestinal lumen there is a level of selectivity. And then so we only see somewhere in the range of 15 to 20 different carotenoids actually in the bloodstream. That's what the eye knows about. If it doesn't get absorbed, it's not going to know anything about it. Next, from that, there is the selectivity that, unlike almost every other tissue in the body, there's extraordinary selectivity of what we put into the retina. As we said, only lutein and zeaxanthin and their metabolites get into the eye. If you look at other tissues, fat, it looks much more like the normal distribution you would see in the bloodstream. Same thing goes for skin. There's only a few that show some selectivity. Among the ones that we can think about and use for trying to think about how this process may occur, the corpus luteum is more selective for xanthophylls. That's another one that tends to take up with a little more selectivity. The prostate tends to like lycopene a little bit more. So there are ways that the body is distributing it into different tissues, and we need to understand how and why this occurs. So, and then there's specificity and selectivity even within the retina, occurring over very, very small areas. And you can see, of course, here, the concentration right in the fovea as calculated is about 100 times what's going on just over here, just a couple millimeters away. So something is different that wants the carotenoids in this area, not over here. There is also chemical selectivity. We talked about the lutein to zeaxanthin and mesozeaxanthin ratio changes. If you look at the bloodstream in the liver, it's more in the range of 3 to 1 to 0 of lutein to zeaxanthin to mesozeaxanthin. In the peripheral retina, that starts changing. We know that there's more interest in the retina in having zeaxanthin in that area. And by the time you get right into the macula, into the foveal pit, the ratio is the classic 1 to 1 to 1. And this work, we heard some of the work done by Bone and Landrum on identifying these carotenoids. I worked about 10 years ago with Fred Katchik, and we really looked at even more systematically to see which other carotenoids could be in there. In addition to the lutein and zeaxanthin and mesozeaxanthin that we've talked about, there are others, and we need to think about what we can learn from them in terms of the functions of carotenoids in the eye and even some of the metabolism. And two of the most notable ones that are there is something called 3-oxolutein. It goes by a couple other different names. And this is the major oxidative metabolite of lutein, probably, and maybe zeaxanthin. And this is found in significant amounts, in the range of 5 to 10% of the carotenoids are like this in the retina. And you can also see this building up in the bloodstream, too. This one is fairly common to be seen out there, and that's not typically in our diet. So that's being formed by oxidative processes. Now, this may go back. You can imagine, or you can not imagine, but there are ways to reduce this and get this back to either lutein or zeaxanthin. And another one that's been kind of neglected that I was thinking about in the last few days that needs to be looked at a little bit more is 3' epilutein. So that's lutein with an inversion at this chiral center. Now that mechanism is very unlikely to happen, just like you don't see zeaxanthin going to mesozeaxanthin. And probably this epilutein is probably, and I think needs to be looked at more, maybe coming from zeaxanthin. And the same process, if there is an enzyme that might take lutein to mesozeaxanthin, could be allowing zeaxanthin going to epilutein. And that needs to be looked at much more, and that may be a way of confirming that pathway exists, because there would be an equilibrium. Zeaxanthin type compounds would be more favored under equilibrium conditions, and there's not a lot of epilutein, but it's definitely there. So how does all this specificity happen? It doesn't, membranes themselves cannot just, because when I first started cannot explain the specificity. When I first started in this field, most people just said, well, they're embedded in the membranes, there's some selectivity. But it's very hard to explain that truly. Membranes generally are not that selective in selecting out a compound and just allowing only two carotenoids to get into the eye. Typically, there has to be some sort of protein mediation allowing this. The most common way this can happen is by binding proteins that specifically interact with a compound and sequester it and draw it into the tissue. And binding proteins are very, very well known in the field, and they've been known in a long time in the carotenoids, and they're well characterized in microorganisms, plants, and invertebrates, the ones where the carotenoids are being made. And the most famous one, of course, is alpha-crustocyanin, which binds astaxanthin and forms the color of lobster shells. And it binds it in such a way that it actually shifts the absorbance so that you get the natural color of lobsters. If you boil the lobster, the crustocyanin is denatured, and you release astaxanthin, which imparts a very strong red color to your boiled lobsters. But what was known for humans and for mammals at all was really nothing, at least in the early 1990s when I was starting to think about this problem. Really all that was known is that there were non-specific binders, things like albumin, beta-lactoglobulin, HDL, and LDL, that seemed to be involved in transport tissues, but they didn't explain the specificity that you would want to have in an ocular carotenoid binding protein. So we hypothesized that they would exist, and so what would they do? Not only could they be involved in the selective uptake of lutein and zeaxanthin, but they could also stabilize it in the cytosol, the cell membrane, and the cytoskeleton. We know the carotenoids are very stable. If you take a retina and explant it, the carotenoids don't, or in a donor eye, the carotenoids don't spread. They stay in their one spot in that tissue. It's very hard, as we all know, to deplete carotenoids from tissues. It takes literally years to put someone on a real carotenoid deficient diet to really see a major sustainable change as we saw with the monkeys. They could be involved in metabolic interconversions. Could be the enzymes involved in converting lutein to mesozeaxanthin. That involves binding. They could be involved in destruction of them within the tissue. Okay, that could get rid of some of the carotenoids in your tissues. Why you may have tissues that are devoid of certain carotenoids. You could imagine, and this may explain in part why you don't see a lot of beta carotene in the retina, is there's a lot of BCO1. There's a lot of that cleavage enzyme, which is another carotenoid binding protein, which was quite happily will cleave beta carotene into two vitamin A molecules, and you'll never see it again. It's not going to be a carotenoid anymore. So that's another thing that could be involved. They could also be involved in other ways of sequestering them in the tissue. For example, in birds, presumably there's an enzyme that esterifies carotenoids and makes carotenoid esters, because their carotenoids in their retina, their xanthophylls, are esterified and put into oil droplets. That's the way they solve the problem of putting carotenoids into specific tissues, even into specific cells. Humans don't do that. We don't see esters really in any tissues except in minor amounts in the skin. And then finally, they may facilitate some of the functions that we're seeing of the carotenoids, facilitating antioxidant and photoprotective action. So to summarize the beginning of my talk, we looked at this problem and we identified in 2004 the first specific carotenoid binding or xanthophyll binding protein found in ocular tissues. And that was obviously about a summary of about 8 years of different work, and of 8 years of work. I'm not going to go into it in detail. I'm just going to summarize what we have. But we identified a protein called GSTP1 that fulfilled all the criteria of being the zeaxanthin binding protein in the human eye. And it was a little bit surprising. This is a protein. This is an example of a protein that has another name, another function, but it does fulfill all the criteria that we would expect to see, including it being in the right tissue, it binding carotenoids, all of the things that we would want to see. And so one of the ways that you prove a protein is a carotenoid binding protein is actually by doing the assays. And I want to summarize or just mention that the way we found GSTP1 was through brute force biochemistry. That's taking literally hundreds of human donor eyes, grinding them up, putting them, extracting the proteins, looking at the proteins that are then interacting with the natural carotenoids, running them on gel filtration columns, on ion exchange columns, through sucrose gradients, and really going all the way to the end till we got a relatively pure preparation that had just a single spot of protein in there, a single protein in there that still was associated with a significant amount of carotenoids. At least we have the yellow marker to follow it through all of this process. And from that, we sequenced it and we got GSTP1. Now, how could we prove that GSTP1 is really the right protein? Well, fortunately for us, GSTP1 was available, and we could do binding assays. Again, this is brute force biochemistry, which I don't encourage many of you to do. It takes a long time to do. And you take your carotenoid, your pure lutein, your pure zeaxanthin, your pure mesozeaxanthin, whatever you're going to work on, and you incubate it with the protein, let it sit overnight, and then do a gentle specific extraction to try to get it away from the protein, and find what's specifically bound to the protein. And we can compare GSTP1 to other carotenoid binding proteins that were known: tubulin, albumin, HDL, LDL, lactoglobulin. And you can see that we get more being specifically bound to the protein, more being bound to GSTP1 as the concentration increases to GSTP1 here, while the others show a lot of noise and very low levels of binding. So that was true when we added 3-RR zeaxanthin, dietary zeaxanthin. It was the same with mesozeaxanthin, shown here. And then when we use lutein, the GSTP1 really doesn't like lutein. You can see it's sitting down there in the noise. And you've got the other ones here. The only one we could even see that had some binding was albumin. That was the only one that we could detect significant binding at all in that area. But you can see albumin looks about the same in each one of these. So it's not very selective and not as strong a binder. And from that, you can calculate binding constants, and we'll discuss that a little bit in a future slide. The other thing you have to show is it in the right tissues in the right areas? And this is Max's one now flipped over because of the way we did this picture here. And we don't have quite as pretty a cross-section through a monkey fovea, but that's shown here. You can see the outer segments. They're stained in green here. And then we used an antibody to GSTP1. And you can see here we get this strip of orange here, reddish orange here. And that's labeling the same area here. That's the Henle fiber layer right in that area. So you can see that our binding protein that the putative binding protein not only did it co-purify, but it's also in the right tissue, and we know that it also binds. So it fulfills the major criteria you would want to see for a binding protein. So we found zeaxanthin's binding protein. What about lutein? And that's really what I want to focus on today. We could tell, doing the same brute force biochemistry, that there must be a lutein binding protein. We hypothesized it. We could partially purify it and do the same binding assays. And indeed, although with a little bit more noise, we could see that in our partially purified systems, there was definitely lutein binding present that was specific more than for mesozeaxanthin and zeaxanthin there. But we hit a roadblock. We essentially hit a dead end. What could we do at this point? Because we couldn't get it pure enough to get a sequence. We couldn't figure out which of these proteins was the one that that we could identify and work with. So we had to get creative, and we had to think about, and I learned at one of the other international carotenoid societies, I think the one in Edinburgh, and Kozo Tsushita is sitting up there, I think. Are you there, Kozo? Yep, he's up there in the balcony there. I wanted to think about, I went to other posters and learned about how other animals or organisms solve this problem. And so I learned about silkworms, and he talked a little bit at his poster, but there are silkworms, and silkworms in their larval stage eat a lot of lutein when they eat their mulberry leaves. And depending on various proteins that we'll be discussing, depending on their protein constituents and how active they are, they either take up the carotenoid and deliver it to their cocoons, in this case lutein, and make yellow cocoons, or if they have various mutations or defects within the system, they don't take up the carotenoid and they make white cocoons. So clearly the silkworm has a process of doing this. And he's made a huge number of advances and really identified the whole pathway, which we'll talk about in a bit. But at that time in Edinburgh, he had identified the major, certainly the one that was of most interest to me, called CBP. And CBP is a carotenoid binding protein that's reasonably specific for lutein. I think it certainly prefers lutein to all other carotenoids, but as we see, it will also bind beta-carotene, some astaxanthin, some other things there. So we took a sort of long shot on this and said, well, could this CBP give us some insights? And sure enough, he had already looked at this a little bit and saw that it was similar. If you do protein homology or genetic homology studies, went through genetic databases, and could this silkworm protein was similar to some human proteins, specifically ones called STAR-D proteins, steroidogenic acute regulatory domain proteins. And it was about 27% homology. Not huge, but if you think about how far we are away phylogenetically from being silkworms to humans, that's not bad. So we took a chance and got an antibody from him and said, does this antibody recognize anything in a primate retina? And this is a monkey retina here. You can see we're right in the fovea here. You can see the cones here. These are the outer segments here, which we know are relatively carotenoid free. We have the inner segments here, and we have the Henle fiber layer here. And at least with this antibody, we see very intense staining in red of the inner segments, and we see some going down into the Henle fiber layer too. And on Western blots, the anti-CBP would indeed identify our reasonably several bands in our reasonably pure protein preparations of a lutein binding protein. So this was encouraging. But which one of these proteins are the ones that we want to look at? Because there are 15 different STAR-D proteins in the human, and that's shown here in this in this tree here that we wanted to look at, and we didn't want to eliminate. We knew that it was closest to STAR-D3, second closest in structure to STAR-D1, but we didn't want to rule out anything, because no one had really looked in the retina. These are all known, of course, to be expressed somewhere in human tissues. But of these 15 different ones, we didn't want to exclude any of them, because all of them shows some homology. So the first thing we did was database searches, and we went in using a full-length sequence of CBP, shown here, and we matched up against all 15 STAR-D proteins. And the ones that are the more red ones are the ones that have the strongest homology. And you can see that STAR-D1 and STAR-D3 are the two that look very, very promising here. The rest of them, you've got STAR-D4 and 5 that are not quite so good, and the rest show some homology, but this is on the very low range. So this at least gives us some guidance which ones we want to look at. Next, we got antibodies to all 15 of these STAR-D proteins. And we looked at to see which ones are even in the retina. And with that, using human macula, human peripheral retina, and human retinal pigment epithelium choroid, and we compared that to a tissue that is similar, mouse retina, that really doesn't want to take up carotenoids. So we hypothesized that we would have something that would be found very strongly in the macula, less so in the peripheral retina. Could be in the RPE choroid if that's the way that it's coming through, but we would expect it to be relatively low or non-existent in the mouse. And again, just as for quality control, we also did liver, which has a lot of different STAR-D proteins. From looking at this, we could see that STAR-D1 looked really pretty grim. It's not found in anywhere in the human retina, in any retinal tissue. It's not even found in the liver. It's found in things like adrenal, and it's actually, interestingly, found in the corpus luteum, which is a carotenoid-rich tissue, so it may have a function elsewhere. But STAR-D3 looked particularly good. As we'll see in some future Western blots, it's stained the macula very, very well on a Western blot, peripheral retina not so well, RPE choroid it was present in, and it wasn't present in the mouse. STAR-D8 was the only other one that even showed any potential, but it was found, it really is not very homologous to CBP. So we didn't concentrate on that very much, and it didn't have macula specificity. And shown here is just the Western blots we obtained, where we had normalized to actin. You can see that human macula, very strong signal here. Human peripheral retina, weak. Mouse, almost non-existent. It was just a faint shadow here. And the same goes with the expression of it. And looking at this, the STAR-D3 is expressed quite readily in the retina of the human. And is it found in the right tissue? And indeed it is. This is shown here, again, another cross-section in a monkey fovea. Again, it's not found in the outer segments where the carotenoids aren't found. It's found in the inner segments and extends actually very nicely into the Henle fiber layer. And if you then pre-absorb using the STAR-D3 protein, the human protein, and to pre-absorb it with the antibody, you see almost no staining at all. So you can see this is highly specific and showing that STAR-D3 fulfills the criteria we'd expect of a binding protein. And shown here is just another kind of nice picture here. Again, you tend to focus on the green, which just shows you where the tissue, where the cones are, because that's a cone-specific antibody. But really look at the red, and you can see how we're really starting to reproduce exactly what was shown in the classic photographs here. So does it bind? That's the next question. Does STAR-D3 bind carotenoids? And we could do it the same method that we had done before, the long-term method of binding and grinding and extracting. But we had upgraded to what's called surface plasmon resonance, which allows us to look at binding in real time on an automated system where we put the protein, the purified protein, on a chip and then pass over a capillary, allows flow over of your ligand, your solubilized carotenoid, which we learned how to do and get reasonably solubilized to do these experiments, and it passes over the chip. And when it binds to the protein, it changes the refractive index here, and you can detect that, and that's linearly associated with the amount bound to the protein. As soon as you then pass if you take away the ligand and pass just solvent through there, the ligand will come off in real time. And you can see a stronger signal with each different concentration. And so we using GSTP1 as the sample here, as the example here, you can see that as we see a stronger surface plasmon resonance response as we increase the amount of zeaxanthin and mesozeaxanthin, while we just see noise here. You can see the specificity of how lutein really doesn't want to bind. So from this, we got some recombinant STAR-D3 and did these experiments. You can just see how purified it was. And you can see here, again, we see a strong response with lutein here, right here, and you can see that as we increase the concentration, the amount goes up. And zeaxanthin does bind, but not very well. The concentrations are much higher here, and if you replot here to generate the more classical binding studies here, you can see that lutein reaches saturation at a reasonable concentration here, while zeaxanthin, while binding slowly, it still has a long way to go. It has a much lower affinity, or not nearly as good affinity for this. And then you can calculate and compare using various proteins, human serum albumin, silkworm CBP, GSTP1, and STAR-D3, and really look at all of these different carotenoids that are of relevance possibly to the eye and seeing what the differences are. Now, looking at these KDs, the lower the number, the better. Okay, so remember that. So you can look at human serum albumin. We're in the micromolar range, which is a relatively high range for this. There's almost no selectivity between these. It'll bind anything. If you look at CBP here, CBP, as expected, really likes lutein here. It has the lowest number here. And there's a pretty big difference between 0.18 and 0.89 and 0.62 for these two, the astaxanthin and beta-carotene. Zeaxanthin is about tenfold lower in terms of its binding constant there. GSTP1, as we found, is the best of all of these, has the strongest binding affinity, and as I said before, it does not distinguish between zeaxanthin and mesozeaxanthin, but it will bind, it is almost exactly between those two, and it's very selective. It does not want to bind astaxanthin, lutein, or beta-carotene. And then finally, what does STAR-D3 do? Well, STAR-D3 isn't quite as good as CBP, but it's still in the excellent range, around the 0.5 micromolar range we think is physiologically very relevant. And you can see that STAR-D3 is actually probably even a little more selective than CBP. You can see here it really has relatively high binding constants with zeaxanthin, mesozeaxanthin, astaxanthin, and beta-carotene. So based on what we know, what I've discussed here is that STAR-D3 does fulfill the three essential criteria for it being the macula's lutein binding protein. It's macula enhanced on Western blots. Its immunohistochemistry localizes it to the right retinal layers, and it selectively binds lutein. And so we're looking at many more binding proteins on this, and we're starting to kind of put them into different classes here, where there's a class 1 that I would propose that has high affinity, KDs lower than 0.6 micromolar, and is really pretty selective. We know for sure that GSTP1, silkworm CBP, and STAR-D3 all fulfill these criteria. I would suspect, although we haven't tested it, crustacyanin probably would do an excellent job with astaxanthin. And there's a whole array of microbial and plant proteins that could be looked at. Class 2 are the ones that are non-selective, medium affinity. So they bind carotenoids, but they really can't distinguish between any of the carotenoids. And we know that human serum albumin, lactoglobulin, we found that STAR-D1, the one that might be involved in the corpus luteum, really doesn't distinguish very much between these. Crystallins, IRBP, which may be a transport protein for carotenoids, and SRB1, one of the scavenger receptors that's been invoked, they bind, but none of them are very good. They're not selective. And so we still need to test things like tubulin, HDL, LDL, SRB2, and CD36, which is a very interesting one that needs to be looked at because it's similar to some of the new silkworm transport proteins. And then finally, of course, there are class 3 ones that have no affinity. If you take avidin, immunoglobulins, denatured proteins, there's no surface plasmon resonance response at all. So where do we go from here? Well, there's still a lot of lessons we can learn from the silkworm, and this is in part from similar to the poster that Kozo presented yesterday. Not only do you have to have a binding sink within the cell, but you need to have the right transport protein. That's what he showed. That's kind of the essence of his poster, is that depending on your CD36-like protein on here, you'll either deliver beta-carotene or maybe lutein. Something is important here. You really need 2 levels of specificity to generate what's probably going on physiologically. So we need to try to figure out how this is going on in a human system. We know it's being delivered here. It's been going into the gut. It has to there's selectivity going on probably by scavenger receptors at the intestinal cell. It goes through the liver. It may make a stop in the liver, blood. Eventually it gets here. Either it goes directly into the retina, or more likely it goes through the retinal pigment epithelium, passes through Bruch's membrane, and comes here. So we're going to be looking at trying to get the whole big picture, how these other proteins are involved in interacting with the system that we're finding of these binding proteins. And then finally, you have to worry about also metabolism. What's breaking down, what's getting rid of the carotenoids from the tissue. So that's going to be what I need to work on over the next few years. So just to summarize what I've talked about with binding proteins, there are specific saturable binding proteins in the human retina, human macula, and other carotenoid-rich tissues. The immunocytochemistry show they're in the right area. GSTP1 is the human macular binding protein for zeaxanthin, and STAR-D3 is the lutein binding protein. And of course, very interestingly, STAR-D proteins play a very prominent and recurring role as lutein binding proteins in really a wide array of organisms. And that we need to, and we still need to figure out what these are doing in AMD. And so we've started some of looking at variants of these and their roles in macular degeneration. And they're not going to be home runs, I can tell you that. They're not going to be like some of the other genes, complement factor H and the HTRA1 that are, have p-values of 10 to the minus 27. These are barely, if at all significant, are low significance. But we need to look at how the diets of people, we need to look at it. AMD is a complex disease. So we're still looking at this more. GSTP1 has been reported by a group in Miami that may have lower expression in AMD eyes. That still needs to be looked at. There's been a recent report that variants of SRB1 may and now CD36 may be associated with AMD risk. And along with Julie Maris, we're starting to look at the association of various variants of both STAR-D3 and GSTP1 and how they may be regulating macular pigment levels in some of her large studies that she's working on. So in the last five or 10 minutes here, if we have time, we still have time here, I'm going to switch gears a little bit and just talk a little bit about measurement of macular pigment distributions in the AREDS2 study. And this, as we've heard, there's many different ways of measuring macular pigment, ranging from the most invasive, which is obviously HPLC, flicker photometry, reflectometry, resonance Raman, and we haven't heard quite so much about autofluorescence attenuation, but that's what I'm going to focus on today. Now, the AREDS2 study we heard is not measuring macular pigment in any in the subjects. It's not part of the protocol, which we think is probably a deficiency. Usually you want to measure kind of response beyond just what's going on in the serum. And even then, they're not even measuring serum carotenoids on more than 10% of the subjects. But we proposed, and other groups proposed, that we should have some sort of measurement system in place, at least at some of the sites. Flicker photometry in the end didn't fly for various reasons and was not used, but we were able to get funding from an outside source to do autofluorescence imaging of our patients. And we specifically wanted to do autofluorescence mainly because it's an imaging-based system, because we knew, having looked at autofluorescence in AMD patients, is that they have a lot of abnormalities in macular pigment. We really wanted to get the big picture, not just have one measurement that you might get from doing a single flicker photometry measurement or from resonance Raman integrated measurements. And so the autofluorescence, very briefly, is essentially using the macular pigment as a filter to attenuate the natural autofluorescence of the pigment epithelium. If you go in with a blue light, or with actually many different wavelengths light, the lipofuscin in the retinal pigment epithelium will fluoresce and emit at long wavelengths, and that's very well known. If you go in with blue light, the blue light will cause this emission very easily, but the macular pigment will attenuate that, and so you'll see a dark spot where the macular pigment is. If there is no macular pigment, you will see much less attenuation. If you similarly do this at a longer wavelength where the macular pigment doesn't absorb, you will not have this attenuation. And from that, you can calculate and image the amount of macular pigment, the amount of macular pigment present. And shown here is our system. You can do this. Based on the Heidelberg Spectralis system, but we have a home-built system using 2 dual lasers and that shines the light on the eye, and you essentially get a picture of the back of the eye, a fluorescence picture. And from that fluorescence picture here, you get the dark spot of the macular pigment centered right at the fovea when you use 488 light, And unlike the Heidelberg spectralis system, we use a 532 light to really get away from where the macular pigment is. And you see almost just a uniform background fluorescence here. And then you can use the same sort of assumptions that people make, that it's probably macular pigment is near zero in the periphery here, that it's, and you do a digital subtraction and corrections. And from that, you can get an image of the macular pigment. And shown here just is the various different distributions you may see. You may see very low macular pigment. You may see just a single spike of macular pigment. You may see these volcano distributions, or you may see a big plateau of these. And from this, you can then measure the optical density at the peak. So in our AREDS2 study, we have 53 subjects enrolled at our site, and we are doing autofluorescence imaging of the macular pigment at every visit. We're also measuring serum carotenoids by HPLC and their skin carotenoid levels. And that's being done by resonance Raman spectroscopy. And shown here is the kind of things we see in the patients that we enrolled. Some are high, medium, low, and some are just a mess here, just all fragmented, probably as you might expect when in very advanced macular degeneration, the macular pigment can get very unevenly distributed. But comparing the AREDS2 patients that had at least reasonable distributions compared to a normal population where we had either low, peaked, or broad patterns, we had a normal population. You can see here that we tended to have more ring-shaped and more head and shoulders or more shouldered ones, more broad, where they have a sharp peak and a little broader area in these AREDS2 patients, at least at baseline. And we could show, as you would want to see, is that when we did see good right-left correlation of macular pigment optical density in our patients at enrollment here, at least if we had both eyes available. And we also found, and I hate to bring this up, but we see it again, macular pigment does decline with age, although it was not, I do have to admit, it was not statistically significant. But it does happen here. The more interesting question is, or the more interesting finding, is the macular pigment was really high in all of these patients here that we enrolled, much higher of what we would consider to be a normal macular pigment level in our unsupplemented population here. And why is that? Does anyone know? It's because they all were taking, 80% of our patients had been taking lutein and zeaxanthin supplements for years and years before joining our study. The washout time is only 30 days for the AREDS2 study. So you have to be, you can see that we have a kind of unique, well-supplemented population. So what did we see? I can only tell you baseline results because the study is still in progress. I'm masked as to who's being treated. But we did see, in terms of we didn't see a strong relationship to MPOD and total serum carotenoids. And remember, these patients had these are very high levels. They were all on beta carotene coming into the study almost. But we even didn't see very much relationship between serum lutein and zeaxanthin and MPOD. But you have to remember that they've been changing their supplements. They've been doing all sorts of things. So we don't see a strong, they probably were not equilibrated yet. What about the skin carotenoid levels? Well, we measured skin carotenoid levels using resonance Raman spectroscopy, and that's shown, this is our device shown here, which, as some of you may know, has been mass marketed to by Pharmanex to the biophotonic scanner. So, but we used ours just because ours is more sensitive than theirs, and that seemed to be helpful. And what did we find is that skin carotenoid Raman counts go up a little bit with age. It's not statistically significant. Again, these patients are very high. These are extraordinarily high levels. These patients have been on a lot of supplements and coming into this study. And the total skin carotenoids does correlate with total serum carotenoids here, but not with total serum lutein and zeaxanthin. And that's because these patients are taking a basic AREDS formulation with a lot of beta carotene. That's really what's driving at least this correlation that we're seeing here. And the lutein and zeaxanthin is not a very good marker for that. And then finally, is there any correlation between what we see in the MPOD levels and the skin carotenoid levels? And the answer is absolutely none. So unfortunately, even though it's a good way of measuring, or easy way of measuring carotenoids, at least in the ARED subjects, it's not going to be a good way to monitor what's going on in the eye. So in conclusion, at baseline, the AREDS 2 subjects exhibited unusually high levels of macular pigment, consistent with the high level of pre-enrollment lutein supplementation. And skin and serum carotenoid measures are not very good surrogate biomarkers for peak macular pigment density. And then I think I'll skip this. Actually, we could talk briefly on this. Do you have time or not for 2 minutes?
Yeah.
Okay. And just telling you where we're going next with imaging is that we're now trying to adapt measures that can be used on infants for measuring macular pigment. Now, macular pigment in infants, you can't use psychophysics. I think that's out. I think what everyone would agree, even the most adamant psychophysicists are going to say that's not going to happen. You can't use autofluorescence because they don't have any autofluorescence. So you can't use that. You can't use Raman because the laser's too bright to be shining in a newborn baby's eyes. But you can use reflectometry because actually a lot of the assumptions of reflectometry where you're worrying about media opacities and all of these other things are gone. And we don't need to worry about that. And we already have a platform we can use called the RetCam, which allows us to image, which is designed to image infants. And you can get an addition to the RetCam, an additional module that gives you very good just blue light instead of the white light. So you can really do blue light reflectometry on these eyes very easily. And shown here is what a blue light reflection image is. And you can then adapt that to a grayscale. You can see the macular pigment very easily in these kids. And you can even do some Fourier transform to get rid of some of the other artifacts in there. And from this, we can really start to see the macular pigment levels in these kids. And what do we know just in our initial ones? We're seeing that the macular pigment is very low, probably zero at birth, if not even lower. Or not even lower, but if you go to younger ages in preemies, it's undetectable also. But you can see it building up now over a period of about 6 to 7 years, and you can see it rising. We're still not sure about these exact levels, but since they're all measured the same way, we can at least see that this trend goes up with age. And now what we're doing is a more complete study looking at skin measurements, serum measurements, all the things that we want to do as they grow. So I'm ready to sum up here. This is, being the keynote speaker, I get 2 minutes to sum up, right? Okay. So what have I learned here and what do we need to do next? And we have a charge, I think, from the Howard Foundation people and the sponsors here to come back in 2 years and have something even more to show for us. So I've just kind of thought of things that I need to do, because I've thought of a lot of new ideas, and I'm sure I'm going to get even more in Poland next week. And so I have assignments for people here too, so be ready on this. Anyone who is a speaker has at least 1 or 2 jobs of what they need to do. So we need to complete and understand the pathways for lutein and zeaxanthin uptake from the diet and into the eye. Clearly I'm involved in that. John Landrum, Wolfgang Schalk, Liz Johnson, see if I can remember everyone who is on here. I think it's very important. We've learned a lot about mesozeaxanthin, and I'm going to take it as a personal challenge. 2 years from now, I hope to be able to tell you what the enzyme is and how it all works. So we need to elucidate the mechanisms for enzymatic or non-enzymatic production of mesozeaxanthin and other metabolites and define their roles. And I think John Nolan, obviously, because he's working on mesozeaxanthin, has to do that. And David Thurman? Okay, I'll get the name right. We, of course, need to define the mechanisms of carotenoid function in the retina and brain. Liz Johnson, Randy Hammond, and who's our B? Richard Bone. Okay, yes, of course, on that. Sorry about that. And we have to continue and improve and utilize the non-invasive methods of carotenoid assessment. And there's many people. I have just a selection of 4 people who should be doing that. And finally, this is a big one, and a lot of us are involved in this. We need to work on clinical studies, the epidemiology, the genetics, get KARMA published, get AREDS 2 published and out there, and that's, there's many of us involved. I'm involved as an AREDS2 investigator. So hopefully we will have, in 2 years, a lot more to say and a lot more information. And I, of course, want to thank, again, everyone who's been in my laboratory and my collaborators, my major collaborators, and the people who have funded me. So thank you.